Let me first thank good afternoon everyone, and let me first thank Professor Alok Gandhi for inviting me to this talk in the conference of pharmaceuticals and nutraceuticals for cancer and other chronic diseases. So today, good afternoon everyone, and let me first thank So, I'll be talking on recent advances in brain targeting using polymerazomes or phagocytes and nanotechnology of brain. With me is Dr. Kritika Koul. She is sitting here. She is also one of the co-authors of this talk. So, first a brief about the nanocarriers for drug applications.
So we are using the mouse only. We have the mouse. Otherwise, if we use this pointer, so it will not go online. So you have to use the pointer only.
Okay. So we must be knowing about liposomes. Liposomes are basically lipid bilayers which are used for drug delivery applications. And many of the FDA approved drugs are being embedded in liposomes for cancer particularly. Then we have polymerazomes. Polymerazomes are polymer-based structures, whereas liposomes are lipid-based structures, right? The difference between them two is, the main difference is that polymerazomes are more stable, robust in comparison to liposomes. You all must be knowing about the polymer micelles, where we have got the hydrophobic core and then we have hydrophilic structure, and we can embed both the types of hydrophilic as well as hydrophobic drugs. And then another one is the dendron. Here you have got a leaf-like dendrons attached to the center core, and these structures which are there, they can in fact take log-minute drugs attached to these at these places. So the drug loading capacity is also enhanced in case of dendron. In polymer and polymerazomes, the drug loading capacity gets very much enhanced because these are the polymer vesicles. So why do they different type of shapes? It's all because of cell assembly of amphiphilic polymers. We get micelles if the ratio of hydrophobic and hydrophilic, we have got most of largest hydrophilic portion and less of hydrophobic portion. Whereas in worm-like micelles, we get equal amount of hydrophobic and hydrophilic structures, whereas in polymerazomes, we have more of hydrophobic and less of hydrophilic structures. So, what are the scientists looking for nanocarriers, which is very important. Nanocarriers have to be safe because when we talk about nanocarriers, these nanostructures should not become like an antigens in the long run where they can create many toxic effects. So the safety is a great concern. Then we talk about biocompatibility. Now I am much older. I have seen many students, many of you are staying here. Whenever they show the slides about cytocompatibility, cytocompatibility is not biocompatibility. So biocompatibility, there is a procedure ISO 10993. Whenever we talk about nanostructures or anything like not nano also, we have to follow ISO 10993. Then in that, there is a huge list of how to evaluate the biocompatibility of any material who goes into the body. Then the last is easily eliminated. The nanostructures are to be eliminated. For example, in 90s, there were a lot of work going on gold nanoparticles, and people used to publish thousands of papers in gold nanoparticles, but later on, the nanoparticle size of gold increases 5 nanometers, it cannot be flushed by the kidneys. So, and then the structure, whether it's circular, whether it is any star shape, that also matters a lot. So, we have to safety is a big concern as long as the nanostructures are concerned. Then, one more important thing is, whatever molecule you take, making the nanostructures, it has to be FDA. Take a new molecule, it will take you 25 years to come to the market. So what are FDA approved molecules like in polymers about the application of nanostructure to brain. So the main cause of memory loss in the brain is either the traumatic brain injuries, diffusal axonal injuries, neurodegeneration, and abuse of drugs. Even if we take the drugs, they also cause neurodegeneration. So the causes of neurodegeneration is the genetic mutations where we have no role to play, but then genetic mutations also have something which the genes we carry forward from our ancestors and great-grand ancestors. Really, a recent the Nobel laureate from Sweden has been a very good about to talk on genomics, where he has told us that even the immune response is totally on the genes which we carry forward from our ancestors. Then inefficiency of cell organelles and accumulation of toxic proteins in the brain that also causes neurodegeneration. So, when you talk about the brain, the hippocampus is where memories and long-time memories are stored and kept. Then, cerebrate gyrus modifies behavior and emotions, and prefrontal cortex is where thoughts of reasoning, planning, argument, doubt, etc. takes place. So hippocampus is the main where the memory loss takes place. So the memory loss and then the failure to store memory and the failure to retrieve it is a very tricky approach. The brain is highly plastic in nature, and we can retrieve the memory also, and the memory can be lost very easily also. So there are not many therapeutics. Coming to the drug molecules, there are the drug molecules based on cholinesterase inhibitors and memantine, where we have seen they worked on prevent the breakdown of acetylcholine. Whereas these are some of these drugs. Rivastigmine is the one which we have compared our the molecule with this drug, and we are seeing that rivastigmine is not working that well. But even the doctors prescribe, because we don't have approvals of many of these plant-based drugs still in the market. Then we have the memantine-based drug. They have got also the side effects, headache, constipation, and many more. And the drug is Namendra XR, which is being given. And still these type of drugs are given. So I'll be talking about the research on brain targeting. The first previous synthesis is derivation of targeted polymerasomes for transport of molecules across blood-brain barrier. Efficacy evaluation of polymerasomes encapsulated with phagocytes and lauric acid alone or in combination against anesthetic agent in primary cultures of mouse cortical neurons using molecular tools. In vivo evaluation of the polymerasomes encapsulated with phagocytes in mouse model of chemically induced amnesia. As you all know that in India, Ayurveda prescribes us Bacopa monida, and Bacopa capsules are to be given up. They prescribe the 2 capsules in the breakfast and 2 capsules in the evening. But mainly people, when they take these capsules, they always say the pain in the stomach. They are not able to take. But none of them it is written that they will get the pain in the stomach. So this Bacopa is a very small herb, and it has got a lot of compounds here, phagocytes A and B, phagocytes, then sterols, then flavonoids and alkaloids. But our focus was to concentrate on phagocytes A and B. And coming to the structure of these phagocytes, they are mainly triterpenoids. Right? All the 3 are triterpenoids and little bit change. Sometimes we have isopropyl group, sometimes you have isopropene group, and sometimes you have simply the double bond. In the students who are, some of you, if you are from chemistry background, see in this case all the glycosides, A glycons are the same. But it is these molecules, this portion, which is always significant for any natural product chemistry. This is the stereochemistry which takes whether the group is axial or it is equatorial. The whole chemistry changes. That's why when these people, the Ayurveda, they give us these drugs, this Brahmi in. I wondered whether they harvest it in the same. For example, if they are done harvesting in August, if they are done harvesting in September or in October, whole chemistry will change. If we are taking Bacopa capsules, I'm not sure whether it will really serve the end purpose. So, coming to this, in our research, Dr. Kritika's PhD was on this BACOP only. So we have demonstrated it has neurogenic potential, neurogenetic properties, acts through tryptophan hydrolase and increases expression of serotonin transporter, increase protein kinase activity, and which plays a key role in learning and memory and helps in the repair of damaged neurons. Although this has been all done where the extract has been taken up, right? And then we worked on another molecule which is lauric acid, which is one of the components in coconut oil, very small molecule but doing pretty good job. This is a coconut oil derived compound, medium chain saturated fatty acids, and potential for killing neuronal hypoglycemia and oxidative stress in AD, PD and epilepsy. And because of its relative small size, they are easier to encapsulate in polymeric nanoparticles and further enhance their transport across the cell membrane. So as we know about blood-brain barrier, it's a barrier which is formed by the tight junctions between the endothelial cells in the capillaries. Many of the students must be knowing this tight junction implies BB its invariable nature. It protects brain, although it protects brain against the trespassing organisms and undesirable molecules and maintains the internal environment in the brain. This barrier also denies entry of drugs for CNS disorders, right? About 98 of the small molecular weight drugs are unable to cross blood-brain barrier. So when the doctor prescribes the drug, it is, I think here it is the 2% which reaches and the rest gets into distributed in our body, and we face nowadays the challenges with the allopathic drugs. So when we started working with bacosides, as it has no ligands, so we encapsulated bacosides in polymerazones, and there are so many different type of ligands you can attack, like lactoferric, transferric, and insulin facilitated receptor mediated transport, and catenized ligands and peptides like albumin can also transfer the brain-blood-brain barrier. But we chose LF, that is lactoferric. It's a single chain iron binding glycoprotein that belongs to the transferrin family. And one of the advantages of lactoferrin to be used as a brain targeting ligand is its low plasma concentration of endogenous LF. So we chose lactoferrin to be, we hooked up to the polymerazones which can act as a ligand, and now comes the role of nanotechnology. As you know, as we spoke, that blood-brain barrier, which is a so tight junctions, and these nanoparticles can be targeted inside and the polymerazones, and it has got the receptors, that transferrin receptors, which can be taken up by endocytosis, and then we have seen in our work that the damaged neuron gets neuronal survival, right? So coming to the polymeric synthesis of polymerazones. See, it's a very simple chemistry, not a very difficult chemistry. We have just taken the one PEG of the molecular weight 2000 Dalton, and then the succinic anhydride, the ring opening polymerization, and then we introduced an SS bond. This SS bond is very important. It's very important for brain as well as very important for in case of cancer, because we have glutathione in axis whenever we have any inflammation, and this SS bond, when it breaks, it can release the drug components into the system. We have done a lot of work as anti-cancerous agents using SS bond, and then in this, then again we do another ring opening polymerization being lactide. And earlier I said we have to use FDA-controlled, FDA-approved molecules, and these lactide and glycolides are all FDA approved. So we played with them, right? And then we got a structure like this, and then we did another ring opening polymerization with caprolactone. Caprolactone has 5CH2, so this was done to have a lot of drug loading capacity. So we want to have polymerazone which can take a lot of drug, because at least 15 to 16% of drug loading has to be there. So in this chain you have SS bond, which SS bond can break up by the glutathione present in the brain. It has an ester link. It can take with the pH, it can be degraded, and the drug can come very easily into the brain, right? So this is the nanoparticle characterization and lactoferrin conjugation. You can see now the polymerisome, core and shell morphology over this polymerisome which we made. This is the AFM showing the pictures of polymerisome, and then this is the CD showing, demonstrating the lactoferrin has been conjugated to the polymerisome, and this is the zeta potential studies where the polymerisome with lactoferrin, the nitrogen content is about 10.72%, whereas without lactoferrin it is 0. We demonstrated that lactoferrin has been conjugated to our polymerisomes. And then we did a side-by-side another study where we synthesize iron nanoparticles with the normal thermal methods which is available in the literature. And we have one set of experiments where only we have iron nanoparticles loading in polymerisomes with lactoferrin, and another set we have bacopa with iron nanoparticles and lactoferrin. And these are just some of the studies which we have done, no need to describe them here, but the more important is we did MRI, magnetic resonance imaging, which was done in mass. And you can see the pre-injection, all these hippocampus, lateral cortex and thalamus, and then we did the study for 60 minutes, 24 hours and 48 hours, and we could find that in pre-injection the values of the iron going inside the different parts of brain was very high, and this is the T2 values in various brain organs, and we demonstrated that iron loaded nanoparticles can easily go to brain, which we demonstrated by MRI, and this study was done in mice models, right? So after moving from this MRI, we moved to optical in vivo imaging, where we use lactoferrin conjugated 900, because for fluorescence we have a fluorescent marker, and here also you can see in the brain the accumulation was very high of our nanoparticles. Then when we're talking about in vitro studies of polymerisome encapsulated with phagocytes and polymerisome encapsulated with lauric acid to eliminate molecular, cellular and epigenetic markers in scolopin-induced primary culture of non-cortical neurons. A comparative study with Patnani as synthetic drug. So,
ma'am, can I just ask a quick question?
Yeah, sure.
So the polymerisome are inserted inside the nanoparticle, or is it a different thing?
Polymerisome is a hollow vesicle like this, and in the drugs are here, and the ligand is here.
That's fine. But what about the iron nanoparticle?
It was inside.
Oh, iron nanoparticle.
Yeah, it was also inside.
So what is the purpose of the iron nanoparticle?
Iron, we wanted to demonstrate whether it is really going to brain. For MRI, you need the iron. Yeah.
Oh, what?
Point crossing blood-brain barrier.
So through receptor mediated endocytosis, we have added the lactoferrin molecules. So these cells in the blood-brain vessel cells, they have the receptor for lactoferrin ion binding ligand. So through receptor mediated endocytosis.
So we have N2A cell culture, and we have primary neural culture, and now this BAN is the bacocytes, and LAN is the lauric acid one, and these are the nanoparticles, and then we have BAN and LAN in 50-50, 1:1 ratio, and then this is the red is the ring. So we have the nanoparticles, all of them loaded separately, and now you can see we did study where we have demonstrated how the neuronal length increases and how the neuronal generation increases. And coming to the second one, so BAN and LAN. This BAN is bacocyte, LAN is lauric acid. Co-administration promoted differentiation of 2:1 positive and 2A cells, which you can see by the relative percentage intensity BAN and LAN. It's very high. And if you look at these pictures, this is lauric acid as such. It does not work that efficient when we make nanoparticles LAN. When BAN and LAN together, and you see the regeneration of the neuronal cells takes place. So, neuroprotection against scolpamine model. We made a scolpamine model. Insert was accomplished with neurogenesis, neutrinogenesis, and neutrine elongation in BAN and LAN co-administered primarily neurons. And look at these pictures, the relative intensity, how they have been changed. Here also it is the length, and here also. What is this treatment? Here I am not able to.
But the dendrite length.
Dendrite length, and it has increased in case of banana. And looking at these pictures, the banana and the dendrite length was increased, which is possible only by the nanoparticles, not by if we are taking the Brahmi as such. So this is the upregulation of BDNF and mRNA levels. Now you can see banana, the BDNF increases very high, whereas in case of RI, that is riboflavin, it is not that much. And then mRNA is again very high, which is a positive side of the research. Then we worked on epigenetic modifying properties of banana, which was evident by altering D and MT and HDAC expression and enzyme activity in primary neurons. We could see DNMT mRNA, it gets decreased, and then we worked on this 3A also, it gets decreased. Then you can see DNMT activity gets decreased. Then again with the different enzymes, 3M, then 5C5, and HDAC also, it gets decreased. So it means it's decreasing the enzymatic activity which are responsible for neurodegeneration in the mice cells. So in conclusions, BAN and LAN are good neuronal health by remarkable efficacy as compared to FDA approved drug rivastigmine. BAN remarkably enhances proliferation of neuronal progenitor better than frequently used rivastigmine. Enhanced proliferative effects of BAN and differentiation of LAN could underline the collective neuroenhancement of BAN and LAN combination. LAN showed profound effect on differentiation and growth of neuronal processes rather than cellular modification. So this was the in vitro studies, combination of BAN and LAN and the rivastigmine. Then coming to the in vivo model, that in vivo we could not get lot many mice, and COVID has started, and pre-COVID also we had not many issues with the animal house. So we did the work with only glycosides. So in the gene expression study was done, this was done using the brain mouse in vivo. And you can see BDNF upregulation, CREB upregulation, R upregulation. So all these proteins which are responsible for the upregulation we are seeing in the in vivo model also. Then looking at the histology, where we have the control, we have sculpamine model, we have the brachiocytes, and we have the band. Where we can see that this is the normal, and then when we sculpamine, this decreased, and in BA it increases, but in with band it is much better. Histology, if you see, it is much better. The CAI, DG, and CA2, it's much better in case of the band. Then the nano formulation of brachiocytes efficaciously reverse sculpamine-induced increase in the expression of chromatin modifying enzymes. So here also in vivo, you can see DNMI, it got significantly decreased. Here also HDAC significantly decreased, and then HDAC C5 decreased. Then again you can see these different type of enzymes, it gets decreased, and CBP, which is the CRIB modifying proteins, and it gets increased. So we did some study on global 5-methylcystone level post-treatment with BAN in imaging model brain, and you can see again a very in comparison to sculpamine, which is decreased, and when the BDNF and R promoter methylation state post-treatment with BAN in imaging mouse model, it gets decreased. Right? So in conclusion, the study demonstrates the efficiency of lactoferrin conjugated polymerisomes as the brain targeting agent, which was evident in MRI and memory loss reversal studies in vivo in the chemically induced memory deficient mice model. In this study, we reported brachiocytes as DNA demethylation and histone acetylation properties in the brain of scolopine treatment emission mice. It was observed that HDAC activity, which was upregulated on sculpamine administration, was restored by BAN. So to summarize, the polymerisomes based on polycaprolactone and polylactic acid increase the viability of drug molecules by effectively crossing the blood-brain barrier. The study demonstrates the efficacy of lactoferrin conjugated polymerazones as the brain targeting agent, which was evident by MRI, and the memory loss reversal studies in vivo in the chemically induced memory deficient mice model. These nano platforms can be utilized for the treatment of other diseases like glaucomas and other pathologies of the brain like AD, PD and any other things which essentially require a targeted approach. So these are some of the papers which has been published, and this is the patent also. The workshop was also attended by Dr. Kritika and the conference presentations. And finally, at last, I would like to acknowledge Dr. KL2 Koul for doing a wonderful piece of work. And we had Dr. Ashish Kumar, he was a postdoc from DST, and he also contributed on epigenetic studies. And epigenetic studies was done in Dr. Arpita Pura. She was an Inspire Fellow in ITIB then, and she helped. The funding we have got all from IT Delhi, no external funding was asked for. So thank you. I hope I finished on time. Do you have any questions?
This amnesic model and Alzheimer is almost same.
Kritika? Kritika is stand up.
So what is difference between these two models?
So actually we have worked with Alzheimer's model, so we have taken the transgenic model. So that study we haven't shown here. So whatever we have taken here is the scopolamine model, which is which causes chemically induced amnesia, and that can be addressed to as transient global amnesia. So it is not specific to Alzheimer's disease, but in most of the studies of Alzheimer's disease, the simplest model which we can take is the scopolamine model. But we have done a specific transgenic study, transgenic animal model studies, where we have shown the lauric acid also in the animals. So that is a different model for Alzheimer's. And in Alzheimer's, we have earlier I have also worked in the stem cell model where we are putting amyloid beta directly injecting into the brain, and then we are rescuing it with the help of stem cells. So I would say the specific model would be the amyloid beta model for Alzheimer's disease. So this is just a mimic of Alzheimer's disease model, because it finally it creates the memory loss. So we are in this study we are only checking the memory loss. The behavioral paradigm which we have studied is only about the memory loss. So we are not checking other plaque detection or that study we haven't done here.
But there also there is a neurodegeneration.
Neurodegeneration is there.
You showed that regenerates.
Yeah, we can rescue it. So we have seen that the neurite length is increasing, the number of neurons is also increasing when we are giving in combination. But we have not specifically targeted the plaques, which are a specific symptom of Alzheimer's disease. So we are not talking about the plaques. In another study, we have taken transgenic animals.
So is the time of administration important with respect to the degree of neurodegeneration which has taken? So once the plaque is made, then I don't think there would be much.
Ma'am, in this is the plaque study is not this one. So so we had this transgenic animal. So they already had the plaque. So in that, I think it's not about the time. So when the time which you're talking about, so that would be when we do through stereotaxis. We have done one model. Through stereotaxis, we inject beta amyloid. So at that time, it will we have to optimize the time, how much time it is taking for the plaque to settle and come as a callus model. So in that case, time is important.
Any further questions?
This is
Thank you very much.
Very good discussion. But you can extend it to Belmont.
Yes, sir. We have done it
You want to ask?
I request Anju ma'am to please participate.
इधर इधर ले आओ